I'm pleased to have Dr. Chris Bohl here with us today. Chris received his Ph.D. from the School of Biological Sciences at the University of Nebraska, where he studied molecular virology. Chris joined Sekisui Xenotech as a member of the product's R&D team, focusing on isolating functional lysosomes from human hepatic tissue, optimizing hepatocyte viability during isolation procedures, and isolation and characterization of hepatic non-parenchymal cells. His distinct test system knowledge, technique, and undeniable expertise led him into the role of Global Technical Support Manager in 2016. Chris has published numerous peer-reviewed journal articles, reviews, book chapters, editorials, interviews, and presentations. It's my pleasure to present to you Dr. Chris Bohl.
Well, thank you, Michael, for that kind introduction. We're going to spend this next 30 minutes or so talking about our CryostaX method and how the expansion of this product is going to help your lab efficiency by giving you extra tools and better hepatocytes that have been pooled, higher activity, and are basically easier to use. But before we get too much into the hepatocytes, I do just want to take a moment to mention how Sekisui Xenotech came to be, or back when it was started, it was just Xenotech. So Xenotech was founded in 1994, and we were an offshoot of an academic lab that was based at one of the local universities here, the University of Kansas Medical Center. Over that 20 years, we grew extensively. And in 2008, we were acquired by Sekisui and became part of the Sekisui family of companies. And this is our 10th year with them, and we're celebrating 10 years of being part of Sekisui. A few years ago, we moved into our current building, which is pictured up top. This is our third move. And in this move, we almost tripled our lab space that was available to us. And we also have more lab space that we can expand into. So we're definitely thinking about the future and where that's going to take us. We've gone from 4 people working in a lab with 1 laptop to right around 130 employees who have worked with over 300 clients around the world. We've also grown our products offering during this time, and we now have our products available worldwide through our distribution network and close partners. So just a quick overview of all of our products that we can offer, or at least a big kind of review. Our product can be grouped into 3 main offerings or pillars of in vitro ADME products. Most people in the field commonly know us for our subcellular fractions, and we have the whole gamut of subcell from all the relevant species, from all the main tissues, liver, lung, kidney, and intestine. And we're well known throughout the industry for our subcell lines. We also have, as Michael mentioned, we've started offering isolated hepatic lysosomes and tritosomes, and those are intended for use for in vitro catabolism assays for biologics. And while we're known, well in the industry for our subcell products, hopefully some of you will start to think of us not only as a microsome group, but also as a hepatocyte CryostaX group. We have our cryopreserved hepatocytes from human and commonly used small animal models. They are currently available in all the usual formats depending on species. That's suspension, attaching individual donors or pools. But we're going to take some time today to explain to you some of the new exciting things that we're going to do with our hepatocyte lines and how we're converting those over to the CryostaX form. We also offer fresh, never cryopreserved hepatocytes for those of you who prefer those for your research. And the last but not least, the last group consists of products that are kind of considered support products for preclinical and in vitro work, such as the immortalized hepatocyte cell line, NAD pH regenerating systems, cell culturing mediums that can be used with our hepatocytes, the research biobank. I want to give a little moment to talk about our custom products group. So basically, if you have anything that we don't sell as a standard product, you can contact us. And this group that runs the custom products group has decades of experience, and they can make almost anything as long as they can acquire the tissue and identify a reasonable protocol. We have lots of really good, strong products that we can offer the field. Another advantage that we like to bring up for our products is that we also use them in our in-house contract studies. So, we have a very good understanding of their use and function. And by using them in our own facilities, this kind of acts like a second tier of QC. So we know that the products that we make and use are of the top quality. So while we have a whole catalog of products, and I can talk to you about them all day if you wanted, but today we're supposed to focus on CryostaX and how they can be used to make your life easier. So one of the first questions that I always get out in the field is, what are CryostaX? So if we were kind of make it the most simple description, is that it's a patented method for cryopreserving hepatocytes in a vial-free form. So vial-free, and in this case, they're formed as pellets. And because of their unique physical properties, it allows a very simple but elegant solution to pooling hepatocytes. For pooling hepatocytes from different donors or different production runs, CryostaX gives us the capability to mix pellets from different production lots into the same vial. And as you'll see in this webinar, it has many advantages over the traditional protocols, which we'll describe here in this webinar. And you can see here that, they really are literally little pellets in a cryopreservation vial. Okay, so now that we've gotten past what is CryostaX, the next big question I usually get is, how is CryostaX going to help me and my group advance my research? And there are many ways we can answer that question, but one of the main advantages for small animal hepatocytes is that by using this pooling technology, we can produce extremely large lot sizes that comprise of many, many tissue donors. So the traditional production methods for producing cryopreserved hepatocytes, especially for small animal models, it's limited to the number of animals that can be perfused at any given time. Each tissue is broken down into individual cells. It's pooled. The hepatocytes are isolated from that pool, and they're resuspended in a cryopreservation slurry. That slurry is then aliquoted in cryovials and cryopreserved. So you can kind of see that you're limited to the number of animals that you can process at any given time. However, using CryostaX, we can essentially make multiple product runs from multiple perfusions and then combine them post-cryopreservation into a single large lot. I ran some quick numbers and we made some estimates, and if we used the rat CryostaX as an example, we could potentially create a single lot with approximately 1,200 vials and over 45 tissue donors. For most companies, this is years and years worth of hepatocytes, and it could be a single lot, so you could get reproducibility over that time period. Another advantageous thing that we're doing with CryostaX is that we're going to simplify the small animal product offering. Instead of multiple formats, such as suspension or attaching, and multiple assured minimal yields per vial, each species is now going to be standardized and getting a standard AMY, or assured minimal yield, of viable cells per vial. Rats will be standardized at 5 million, mice at 4 million, and our monkey product is still to be determined, but we expect it to fall somewhere very close to those other AMYs. Another thing that will simplify things for our customers and end users is that the rodent CryostaX will have a single format use. And basically what this is that the CryostaX can be used for suspension or attaching assays, and the hepatocytes will come with the same characterization that you've come to expect from Xenotech. So also with CryostaX, because of the unique properties, they also have a simplified thaw procedure. Okay, so basically there's no more water baths in the lab. There's no more thawing vials in that water bath. As seen in the video, you simply pull the vial from the vapor phase, pour the physical pellets or cells into the pre-warmed thaw tube, and you rock it back and forth. The cells thaw more quickly and more evenly, and it takes out a source of variability in the thaw process. Once the cells are thawed, they're spun down like normal, resuspended in incubation or plating media, and you're ready to go on to the next step. And for those of you that already use our hepatocytes, not to worry, this conversion to the CryostaX format, you're still going to get the same great quality they're just easier to use. So in this slide, you can compare a data sheet from a recent vial lot, which is the traditional lot, versus one of our new CryostaX lots, and they basically perform the same. Another one of the advantages of CryostaX is that you basically can get human pools of hepatocytes that have less cryo-injury and thus higher activity compared to the methods that use 2 cryopreservation steps to pool hepatocytes. Much like what I showed with the animal hepatocyte products, human hepatocyte isolations are also limited to the amount of tissue that can be procured and processed at any given time. The traditional methods call for individual donors to be processed and cryopreserved as individual donor lots. But if you wanted a pooled product, those individuals would have to be thawed, pooled, realiquoted, and recryopreserved, putting them through a second cryopreservation step. Excuse me. This second cryopreservation step naturally increased the cryo-injury of those hepatocytes. Now, cryo-injury in hepatocytes is real and can have a very significant effect on the quality of the cells. In this chart here, our data shows that each cryopreservation leads to a decrease in overall metabolic activity. This chart very clearly shows that after every subsequent cryopreservation, the average metabolic activity of individual donors decreases. So if you need a pool for your work, obviously the more cryo injury you have, the less quality of cells that you'll be able to receive and use. The same phenotype is also replicated in pooled human hepatocytes. So the previous graph was showing individuals, the average activity decrease over multiple cryo injuries. And this also has an effect on the pools. So this also is one of the reasons why CryostaX is so advantageous for the use in labs, in that we minimize cryo injury while maximizing the amount of activity available in those cells. By using pooled CryostaX, you're able to use hepatocytes that have limited cryo injury, as the CryostaX system allows pooling of a product that has only been cryopreserved once, and that's an industry first for pooling products. And you can basically avoid the negative effects of multiple cryopreservation steps. Also, because of the ease of pooling, we can customize the pool if we have the individual donors that match our criteria. Most times we can have the custom pool in our hands within 3 to 4 business days. And listed below are just some of the examples of the custom pools that we've produced in the past. So sometimes maybe a gender-specific pool is made because the compound is targeted towards a drug, or excuse me, a disease state that's maybe in one gender or the other. We can also custom pool based on ethnicity, high or low specific activity, number of donors can be changed. And another thing that is really advantageous to the CryostaX system is that we can very easily alter the number of AMY in that vial. So if you want an individual, but you only need 1 million cells, we can very easily do that. Whereas if you had the traditional vial, you would have to do a 2-step thaw, basically. So you would have to thaw the cells, re-isolate them, re-cryopreserve them, and then you'd be faced with the second cryo injury hit. So the CryostaX technology allows for easy customization. So if you're interested in a custom request, you would contact us through our website, and myself or another scientist will search through our fully characterized individual donor inventory and create a pool that best fits your needs. So all of our CryostaX individual donors are fully characterized. We have available to the customer medical histories from the hospital prior to donation, and all the activities commonly used for characterization. So in the past, we have spent considerable time qualifying our pooling process, and to be able to show that what we put in is basically what you're going to get out, and that the theoretical average of all of the individual donors ends up being very close to the actual measured average activity of the pool. So another way to show this data and to convince people that the pooling process is a good way to go about pooling hepatocytes is to show the data from each individual that has been used in the pool. And with this, we can show how their theoretical average activities compare well to the actual characterized activities of the pool. If you look across the individual donor activities, you can identify CYP activities that have a wide range across the donors. For instance, if you look at 2C8, 2C9, 2C19, and UGT activity, these are all good examples of activities that have wide variation. But when you look at the pool's theoretical average, it's in good agreement with the measured activity of the pool. And this strongly suggests that one donor does not over or under contribute to the pool's average activity. Through this qualifying work of our pooling technique, we've also shown that CryostaX hepatocytes keep their inducibility, and that these are very good pools that can be used for induction screening. We also have data, and we presented this at meetings, that the pooled CryostaX attaching human hepatocytes can also be used in examining intrinsic clearance rates of compounds that have been classified as low turnover or microsome stable. And this is by taking advantage of the CryostaX ability to pool, which helps average out inherent individual donor variation combined with the hepatocytes that have minimal cryo injury. So we kind of get the best of both worlds. We can limit donor variation, but keep maximal activity. And that's kind of key to being able to get metabolism of some of these really stable compounds. And while there are other methods out there to examine low turnover compounds, each one has their own advantages and disadvantages. But over the course of time, we have found that the CryostaX data, it's in a good agreement with the other test systems. And overall, the CryostaX system is generally easier to use and more cost-effective than the other systems available. So just to reiterate the advantages of human CryostaX and CryostaX in general, is that they're available in multiple formats. You can have them as individual donors with custom assured minimal yields. So if 5 million is too many, we can turn that down to 1 million, 2 million, 3 million, or anything in between. These are the only available human pooled hepatocytes on the market to be subjected to only a single cryopreservation process. Because of the CryostaX technology, they're easily customizable. You just give us a call, let us know what you need, and we'll be happy to help you out. And we've shown through multiple experiments that these are effective for evaluating low, mid, and high clearance drugs. And the platable hepatocytes are suitable for uptake studies and CYP induction screening. And basically, as we roll out the expanded CryostaX product line, things are simpler, they're easier to use, and we think that they're going to help efficiency in your lab. We also offer all of the support media for use with our hepatocytes. We strongly recommend using our media as we use our own media for characterization of the hepatocytes. And use of our media is the best way to reproduce the results given in the lot-specific data sheet. Using our media and our approved protocol, we can be much more helpful with technical support if we know exactly what you've done and what's in the media. So hopefully this has given you kind of a good idea of what CryostaX is, how it's easier to use, how it's more efficient, and how we're going to be able to help you continue work more efficiently in your own lab. So for the CryostaX animal hepatocytes, we currently have the rat and the mouse, and soon to have monkey. We have the CryostaX in suspension or individual donors. You can have those as a platable format as individual donors or pooled. And while I didn't talk about it, we've also used this technology to create a product called the genome human hepatocytes. And what we have here is a 3 individual pool of hepatocytes that we've identified specific CYPs within certain metabolic genes. And these CYPs that we've identified have been described in the literature to be directly involved with affecting those CYPs or metabolic activities in the literature. So we can offer a high activity, a moderate activity, and a low activity of mini SIPs. So hopefully I've convinced you that the CryostaX are a good thing, and that you hopefully would like to get them into your hands and give them a try. If you have any questions, I'd be more than happy to answer them for you. My email is at the very beginning. If you have a question that comes up later, please email me. I'm more than happy to sit down and respond, or even give you a call to chat. So just let me know. I'm available for help anytime.
All right, thank you, Chris. Information about our test systems and services, other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter.